Hello, I'm Ana Casas. I'm the Work Package 2 leader within the H2020 Repo Trial program. Today, I want to give you some insights regarding the concept of network pharmacology and how in silico predictions could lead to real patient therapies. And let's start from the beginning. It's definitely not a secret that the translation from preclinical research to the clinics is still extremely complicated. So less than 1% of all tested drugs finally reach the clinics, which is almost nothing. So this is indeed a huge medical and social problem. So the question here is, what exactly is failing in clinical translation? Well, actually, if we have a look at the numbers in detail, we see that preclinical and phase 1 trials are finished primarily due to safety reasons, as you can see here. However, if we focus on phase 2 trials, they fail primarily due to efficacy reasons, which means that the drug simply does not do what it's supposed to do. So based on this situation, we believe that it's definitely time for a change. That's why we want to introduce the concept of network pharmacology. So in current therapies, it is common to first identify different symptoms related, of course, to different targets, and therefore treat them with 1, 2, or 3 completely unrelated drugs. And this is what we could consider classical combination therapy. But here, we want to propose something different. We want to propose network pharmacology, where the focus is centered on the causal mechanism underlying the disease. In this case, we will target a complete disease module, of course, including different targets, that will be treated using mechanistically related drugs. Therefore, our strategy should not be confused with a common combination therapy approach where the mechanism of the disease is not fully taken into account. Then, network pharmacology suggests that by targeting different proteins within the same pathomechanism, you should expect not only an additive effect, it's the one that you see here, but a full synergistic effect. So if you're looking for synergy, you of course can maximally reduce the dose and start using subthreshold doses. And related to that, you could maximally reduce potential side effects. So network pharmacology will give you the option to have minimum side effects with a maximum full synergistic therapy. At the same time, our approach to different disease indications also follows a network-based strategy. That's why I'm presenting here the DISOSOM, the network of all human diseases. In each node, we have different disease indications, and the edges connecting them are the genes that they share. So if you put together all human genes and all risk genes, you end up with this network. In some cases, like for example here in light blue, we have an homochromatic cluster, including different types of cancers. Or the same here in red, where you have different neurological diseases. However, if we have a look at the network as a whole, the majority of the network is multicolor, which means that diseases that you would put in different categories, they mechanistically belong together. Therefore, we suggest to move far from an organ-based approach and focus on the underlying cause, on the underlying mechanism causing the disease. So by doing that, we identified a cluster of diseases sharing a common underlying mechanism, the impairment of reactive oxygen species formation and cyclic GMP signaling. This is what we called the ROS cyclic GMP disease cluster. So diseases that in principle had nothing in common, like for example, you have here different cardiovascular diseases, like atherosclerosis, heart failure, but you also saw some metabolic diseases like diabetes and obesity, and some neurological diseases like Alzheimer's, Parkinson's, and stroke. So due to the unmet medical need previous experience with in vivo and in vitro models, we decided to focus on a stroke. So now it's moment to actually give you some context, and it's important to mention that a stroke is currently the first cause of death and first cause of disability at the moment. With no neuroprotective therapy available, no specific therapy for all those patients suffering from post-stroke disabilities, which are around 45%, and no specific diagnostic biomarker. All of that part of a long-lasting translational crisis. In fact, more than 1,000 experimental treatments have been tested in a stroke. And what was the success rate over the last decades? Actually, none. Nothing worked so far. And that's why almost all pharma companies have completely withdrawn from stroke research. On top of that, it's important to mention that all failed strategies were based on single compounds targeting single proteins, making evidence that the one target, one drug approach is simply not working, at least in a stroke. Then we decided to follow a network pharmacology strategy here. So we started with an already well-validated target in a stroke, NADPH oxidase 4, also called NOX4, as a seed-validated target. The one here in the center. Then we found out that the closest targets linked to it through both metabolites and protein-protein interaction was different NO synthase. So specifically NO synthase 1, 2, and 3. The ones actually located here around. At this point, we moved to the lab and we used an in vivo model of brain ischemia. So to induce ischemic stroke, a silicon filament is inserted in the middle cerebral artery to actually create a complete occlusion. So in detail, permanent sutures are located in the carotid artery and in the sternal-carotid artery, while the filament is inserted and located through the artery until it reaches the MCA. The time of occlusion will depend on the severity of the model. So for an acute, highly severe model, the filament will be in place during 60 minutes, so 1 hour. In this scenario, animals will be sacrificed maximally 24 hours after reperfusion, so after the surgery. However, in case we want to evaluate post-stroke readouts in later time points, the occlusion could also last instead of 1 hour, 40 minutes or 30 minutes. So in this case, animals will be able to survive for months after the intervention. So using this model, we defined a network pharmacology therapy where we combined NO synthase inhibitors and NADPH inhibitors. So we combined 2 different drugs. And this is what we saw. We saw a reduction in infarct volume at different time points, so not only 1 hour, but also 3 hours after stroke. Also in aged animals, not only in middle or adult animals, but also in aged ones, and both in a transient model and in a permanent model of brain ischemia. These results were in line with the improvement of neuromotor functioning, as you can see here with this battery of tests. Also the reduction of blood-brain barrier leakage and the reduction of ROS formation, of reactive oxygen species formation. However, we are not only focusing on neurovascular diseases, but we are also working on other indications part of the same disease cluster. So lately, we have been also working on heart failure. In this case, in collaboration with the University of Halle in Germany, we established a mouse model of heart failure by administering 4 weeks of angiotensin II. So after the model has been established, these animals were treated with a network pharmacology therapy followed by the assessment of different readouts, including echocardiography, blood pressure, the running well activity, and also different histology assessments. We obtained really promising data, which served as the basis for a clinical trial phase 2 currently ongoing, where we are treating patients suffering from heart failure with preserved ejection fraction. Finally, before I finish my presentation, I want to introduce our recently developed preclinicaltrials.org platform. This preclinical repository gives you 3 key functionalities or 3 key main options. So it gives you the possibility of accessing to material from already performed animal experiments, like for example, tissue and blood, the deposition of unpublished negative data, and the inclusion of raw data extracted from already published studies. In fact, if we want to deposit unpublished data or raw data from already published studies, this platform should be flexible enough to be able to allow you to classify the content and include as much as possible as the study that you perform. As the final slide, I just want to show you how diving into the dysosome could look like, seeing not only the different disease indications, but also the genes connected them. So at this point, I just want to thank you, of course, for reaching this point of the lecture. And here you have some interesting references in the topic, just in case you want to check them. Thanks a lot. And of course, we can always talk by email or any other way of contact. Thanks.